Hey everyone, we're now here to talk for our third episode, right, Uza, of Sambai Speaks. I'm Ricky Fairley, and today we are talking about our work package 3 about exposomics. And I'm sure you don't know what that word means, so we're going to spend the next few minutes talking about what that means and what that means to our research. But again, Sambai is our research project that we're doing in, we're doing it now in the US, Canada, the UK, Ghana, Nigeria, South Africa, Ethiopia, and Kenya. And we are doing this 5-year global research project and really trying to understand the disparities with cancer. And we're studying breast cancer, prostate cancer, and pancreatic cancer. But today we have our amazing scientists on our work package 3 team. We're going to really just talk about the work that they're doing and what it's contributing to the bigger picture of the project. And this was the hardest one for me to understand just because it was so foreign to me, but they're going to break it down like we're 5-year-olds, like I love, and in words we can spell. And I'm going to let them introduce yourselves. Dr. Miller, do you want to introduce your team?
So I am Gary Miller. I'm a professor at Columbia University in the Department of Environmental Health Sciences. And I do research on the exposome, which I'll explain to you, and I'll let Kam-Meng introduce herself.
Hi, I'm Kam-Meng Tchou-Wong. I'm an executive director in Dr. Miller's Weinstein's for Research Strategy and Innovation at Columbia Mailman School of Public Health. Prior to my joining and working supporting Dr. Miller, I was a cancer biologist in training. I got a PhD in Princeton, and I study cancer, actually, in Epstein-Barr virus that is related to Burkitt's lymphoma and nasopharyngeal carcinoma. And subsequently, I study lung cancer early detection and biomarker. And now I'm very excited to join and support Sambai studying the important breast cancer and its etiology and disparity.
Great to have you, and use some really big words, but I know it's all going to come out in the wash, and you're going to help us with that. And then we have Danielle. I'll introduce her. So on every work package team of scientists, we have a patient advocate, which is such a great thing. And actually, I'm pretty sure this is the first time that a Cancer Grand Challenge, a research study like this, has actually had patient advocates like me and Uza and Danielle involved in the biological science, in the science. And so we're so happy to be here. And so Danielle is our advocate on our team. So
yes, I am a 24-year breast cancer survivor. I was diagnosed with invasive carcinoma, stage 2. And later on, after my diagnosis, I was genetically tested and I found that I had BRCA2. So those are my background with this, but also being from Louisiana, I live in the area of New Orleans, better known as Cancer Alley. So it has environmental situations where your genes could have been, what's that word? Changed.
Change.
Yeah, change. That's a good word too. By the chemicals that were being used. And so I'm blessed to be a part of Work Package 2. I mean, I came in at a certain time when y'all needed someone to lead, to be a part of that as a patient advocate. So I feel blessed to be a part of the Exposomics work. Also, I have my MPH, and I love the GI work that we did, GIS work that I did when I was getting my MPH. So I'm just excited to be a part of this team and actually be the patient advocate for it. So yes.
Awesome. So okay, so Dr. Gary, what exactly are you studying? And I love that your background is really with Alzheimer's, more patients more than cancer, right? So it's kind of even a little bit of a new area for you, but can you just explain what is exposomics? What does that mean?
So I think Danielle gave a perfect summary here. She talked about how she has a genetic risk factor for breast cancer. But just because you have a genetic risk factor doesn't mean you're going to get the disease. It's a risk factor. And all human diseases are usually a combination of our genes and environment. So the field of genetics studies those genes and how they do that. And when they look at all your genes, they call that genomics. So genomics just means you're looking at all the genes in that individual, or in a population, whatever it be. So the concept of the exposome was proposed by a very famous cancer epidemiologist, Chris Wilde, about 20 years ago when the Human Genome Project had been taking off. And he says, we had to have like an environmental complement to the genome. And he coined
What's a genome? What's a genome?
So the genome is all the genes in your body.
Okay.
That's the whole complement of that. So genomics is studying all the genes.
Okay.
And so he suggested this idea of having a complement to the genome, and he called it the exposome. So exposomics is like the genomics part of that. We're looking at all the environmental exposures that people face and throughout their entire life. And so the idea was to have that environmental balance to the genes. And so you might say, wow, how do you measure all those things?
So let's break that down a little bit more. So environmental things that could be affecting you could be where you live, the climate, right? Or could it be like even stuff that you put on your body, like products, or like being around, to Danielle's point, being in an area where there's a lot of chemicals in the soil, or like to me, like even Flint, Michigan could be something like where the water is bad, right? Right.
Right. So it's all the things that you bring into your body, whether you're drinking it through water or ingesting it through your diet or through, healthcare products like, beauty products that you put on your skin or your hair. It's all of those complex exposures. It even includes things like temperature and humidity and radiation, exposure to things like pollen, so even allergens and things all fall into these exposures that aren't controlled just by your genes, right? The idea is these non-genetic factors that are doing this. And so the idea is that when you think about all of those things we get exposed to, we wanted to see how can we measure those things? So there's some things that all we can do is estimate the exposure. So for example, like the amount of sunlight you get. Well, that's something that's measurable if you had some sort of sensor on you. You could look at how much noise someone was exposed to. And then if you know where people live, like the geospatial thing that Danielle, geospatial just means you're looking at the geography in space of where you are. And if you've ever seen like a map of the United States that shows the pollution, like air pollution across the United States, you can link up where people live to what these other factors are. What my research group is focused on is because many of those things we just talked about, like food preservatives or pesticides or metals you're exposed to, all these sort of small molecules, they're chemicals. They're small chemicals that are in your body. Essentially, all the things going through your blood are all small chemicals. That's what they are. Is using techniques to measure as many of those chemicals as possible. So we do it by taking blood from people and analyzing all the things we can analyze in it. So we're just trying to measure it. There's hundreds of thousands of things we see in the blood, and we're trying to figure out what all of those things are.
Like a blood test, like a blood test on steroids, so everything that could possibly be.
Exactly. Like you go to the doctor and you do clinical chemistry and they show you 40 things. We're trying to show you 100,000 things.
Wow. Wow. So how do you do that though? That's a lot of work.
So again, when you think about when you go to the doctor and you give a blood sample and they run things like cholesterol or creatinine, for their testing, kidney function or liver function, they're using techniques to measure those small chemicals. The fancy term for a lot of that is called mass spectrometry. So you may remember doing light spectrometry in graduate school, in high school sort of thing, where you looked at light in a cuvette sort of stuff.
Yeah.
This is
I
just,
was never good at that stuff. I was never good at that.
it's just fancier versions of it. It's just like equipment that, like if you may remember like using a microscope when you were a kid, and they have these electron microscopes that can look at like strands of DNA. It's still microscopy, like they're still using microscopes. They're just really fancy ones that you use in a research laboratory. It's the same thing as the chemical analysis. We're not using the one you used in elementary school. We're using the ones that cost $1 million each sort of equipment.
Okay.
The idea they can just measure so much more because of how sensitive they are to do that.
So you basically are going to measure a lot of blood. And for our whole study, we're going to try to have 40,000 people in our study, right? Across all the countries. So that's a lot of blood for one thing. But so you're just going to kind of look for commonalities, look for trends, look for, like, what are we looking for? How do you even know? Will you find that out what you're looking for once you look at it?
It's D, all of the above. Like we're
Okay.
looking at this in many different ways. So if we're looking like in a routine study we would do, we may look at a population of people that developed a disease like type 2 diabetes. And so you would look at people that are the same age that don't have it, and then people that develop diabetes. And you can just compare those two groups and see what is different between them. You would think if you have people that aren't treated at the time, you'd see higher sugar in the people with diabetes because they can't control the blood sugar level.
Right.
Be lower. And so, but when that happens, that sets off all sorts of other things. And so it's not just the blood sugar, but all the systems that the sugar is feeding into. And so with something like cancer is that you have, the idea is that when you think about the process of cancer, you may be exposed to some chemical that initiates the cancer, that causes the initial cells having a mutation and growing faster. And then the disease progresses in some way. And then you might start getting therapy and recovering from that. And all of those steps can be affected by your environment. So if you have a really poor diet, you may be more likely to have that first event. You may progress faster because of the poor diet, and you may not respond as well to the therapy because of the poor diet.
Right.
But when you say poor diet, that's the chemicals you bring into your body. It's the combination of the good and bad chemicals.
Right.
And so that's what we're measuring. We can measure these chemicals. And so the idea is that we're not just saying that a person ate a poor diet and they lived close to the highway. We're measuring the chemicals that are causing the problems. So just because you're near a highway, what does that mean, right? So the idea is that there's going to be more pollution. There's going to be diesel exhaust, gasoline exhaust. There could be other things that just come off the equipment as it heats up in your car. But those chemicals, to have an effect on you, have to get in your body. And so the idea, by measuring it in people's body, we're not just estimating it from where they live, which we can also do, but we're actually measuring it in that individual.
This is
Wow.
great because it really gives us a laser focus on
Yeah.
each participant in this study. And the more you know, the more you can take care of yourself. So if you really have all of those specifics on your environment, on your food, and how all those things are connected, it'll only help you take better care of yourself. So really excited about this.
And I think, one of the things that we've talked about doing as we go forward is that when people make what they think are healthy changes in their lifestyle, how do you know that? How do you know you actually did that? Like you could wait a while to say, oh, I lost some weight or my cholesterol came down.
Right.
But if you improve that your chemistry changes in a positive way.
Yeah.
And these techniques we're using, like the idea you could say if someone undergoes something where, let's say, for example, that a factory nearby improved their pollution control, you should be able to measure that not just in the air, but actually in the people that live here nearby.
In their blood. Wow.
So this is like, I'm in a whole food plant-based group, and we talk about a lot of people to mirror their diet in that way. So what you're saying is just because we're saying it basically lowers your cancer risk, you could tell in someone's body whether the cancer risk has really been lowered because of the foods that they're eating.
You would, the idea of that risk going down, like if you know that these certain chemicals in your body are risk factors for cancer, yes, you could measure the reduction of those things. So a good example is that if someone was concerned about pesticide exposure, which is an interesting thing, like we want to have as low an exposure as possible to chemicals like that. But if we had no pesticides, our malaria cases would be 100 times higher, right? So the idea is you need to use some of these things to control bad insects. But if there's been studies where people like go on organic diets and you do see less pesticides in their bloodstream, are they healthier because of that? That question is a bit harder to answer, but you can at least tell that you reduce that exposure, which is one of your concerns. And so the idea is that you can reduce the exposures, and in general, the fewer chemicals we're exposed to is better, unless that chemical is a medication you're taking to control your blood pressure or to treat your cancer, right? I mean, there's some of these chemicals we like because they have very positive actions.
Right, they're doing something for us. So this is pretty fascinating in terms of the scope of this project, because I'm just thinking too, like in the US, we have, a good diet, a bad diet. Like Danielle's really healthy because she eats, vegan. I eat ice cream every day. I just had a root beer float before we got on the Zoom. But I mean, there's so many people who eat badly here. But if I was in Ghana, I would have a totally different diet, right? That could impact my blood cells or what you're looking for, right? So it's going to vary by country. It's going to vary by culture, right? And are we looking at all that too? I'm sure we are. Just,
Right. I think this is like a big part. What I love about the Sambai project is that we're not just doing exposomics. We're not just doing genomics. We're not just doing social determinants. We're doing all of them.
All the things.
And so the idea is that when you think about the social factors that impact our health, like what are those factors? What do they do to the individual? Often there are things that explain higher levels of stress. The social determinants might affect where someone lives, which could affect their exposure to pollution. It could affect their access to healthy food or to proper health care. But all of those things, like if those things are going to impact our health, they have to go through our biology.
Right.
So by taking these blood samples, we can see that. So if we say, from the study, let's say, for example, in one of the countries in Africa, they do a lot of electronic waste recycling. A lot of that is like we send a lot of our old computers and TVs to Africa to do the recycling. So the people that live closer to those are going to have more exposures to the chemicals that are in that process. And so you could see, like, depending on how far people lived away from that, does it change their exposure to these different types of chemicals? But exactly right. It's going to change on different populations in different countries, in different regions of the countries. We know it changed, like within New York City, it's not just the boroughs. I mean, street by street, you can have a big change in exposure within a city.
Right. Gosh, it's going to make us like figure out where we want to live. Right. Like, where is the ideal place to live based on the exposures around you?
So I know you said Christopher Weil created this and it was 20 years ago, but how relevant has this work been around? I know he created it 20 years ago. Has it been around for 20 years where people have been really paying attention to it? Or has it just basically really started coming more into focus now at this moment?
So, excellent question. So Chris Weil coined the term in 2005, but really very little happened for about 10 years. And part of the problem there was that the tools we needed to measure hundreds to thousands of things hadn't been developed. So people started thinking about it conceptually, but they didn't have the right techniques to do it. And so that's what's happened over the last 5 to 10 years, is that the advances in chemistry have gone so far where now we can do much more of this. And I realize that there's really bad glare on my camera right now.
I know, I see
The other
it, I know.
reason too, like I have a standing desk and I can move around a little bit, but it's
We can see you.
I can't seem to avoid it.
See you. So, I'm just thinking about how we how we relate this back to disparities. So there you are. You came back.
I'm lowering my desk.
Okay, that went away. That's all right.
So,
So go ahead.
yeah, so thinking about the disparities is I'm sure you know Linda Villarosa. I was just at a seminar of hers today, wrote the book Under the Skin, where she's talking about a lot of these health disparities. And so when you think about living in a disadvantaged area, again, it's not like a mystery of how these things happen, right? It has to get under your skin. It has to change your biology. And so if you're trying to determine, like I think the example of COVID was great in New York City. There were areas in New York City where the COVID rates were so much higher, the outcomes were so much worse, and we're all in the same city. There's hospitals around all these different places. I can guarantee you that wasn't genetics causing that, right? It was the neighborhoods that people lived in. It was the levels of pollution they were exposed to. It was the lack of medical care they had access to. It wasn't the genetics. Now, not to say there weren't some genetics involved in overall vulnerability,
Right.
but when you saw it in that acute phase, that was all disparities, right? That was health disparities because you could see the geographical distribution, and that's the sort of thing you're looking at, because if it's due to living closer to that factory or highway, there should be signals of those exposures. There should be evidence of higher stress levels because of the level of crime in that area or something. And so the idea is you're measuring the downstream effects of the drivers of health disparities, and you're measuring it in this sort of like biochemical level.
Wow. It's like one of Dr. Lauren McCullough's facts from her Emory study about the social determinants of health. She says that Black women who live in a high socioeconomic area, neighborhood, still have 126% higher mortality rate of breast cancer than the white women in the same neighborhood. And I'm thinking, well, how is that possible? But she said even that could be basically based on racism, because they don't feel comfortable, or the stress of living in that neighborhood with people that don't look like them causes a stress on their bodies that could have an impact on their cancer.
Exactly. And that's like the sort of example when you see those situations where you're like, what, like they're in the same neighborhood. So you could actually demonstrate that their exposure to pollution should be the same because of the neighborhood, but how they handle it could be very different. Right. And so the idea is it may not change the levels you see in the blood, but the pathways that are activated in your biology could be heightened because of that, because of like the increased stress level. Like this chronic stress is not good for our health. And chronic stress, like we think about work-related stress or financial-related stress, but it could also be more social structures like racism causing that stress. Those are all real, they cause changes in your biology.
Right. I know I actually did a clinical trial at Georgetown. I guess it's been 2 years now, where they measured my stress level as a survivor. They were measuring survivor stress. And I had to go quarterly and give blood. And basically they just did like a deep dive analysis, like a 200-page questionnaire survey, kind of what my daily activities. But we don't have the results of that. So they just closed the study like a couple months ago. So I can't wait to see the results of how, what they said about, and I think the women were predominantly in the DC area. So we'll see. And we know breast cancer has a horrible stronghold in this area.
Yeah, it's interesting when you think about like controlling stress. I used to always think that things like yoga were silly, But when you see the data on how these methods to reduce stress actually help pull those stress levels back down, like we know throughout history in different cultures that there's meditation sort of skills that people use, and it has a biological change. And so if these negative things can increase your stress, there's positive things you can do to decrease your stress.
Yeah. I
Oh,
used to
stress-
that was for women. You know,
Stress
so-
also affects the immune system, and
Yeah.
cancer is very important, right? And immunity is whether suppressing the cancer growth in cancer immunity or general immune health is very important. So that chronic stress definitely is affecting your recovery, right? Your treatment response and your cancer. Yeah.
I really do believe my cancer was caused by stress, you know, and I think even a lot of it was self-induced, I think, but I really do. Because even before I had cancer, I had I always had something, a sinus infection, a yeast infection, something, a runny nose. I always had some physical thing, and then I had cancer. But I'm sure it was because I say I don't believe in stress. Stress is for wimps. It's a cop out. What are you going to do? Not go to work, not get the kid, not do your stuff.
I say the same thing when I had blood clots. So I've had blood clots twice. And the first one they said it was a side effect of my cancer. So I said,
Wow.
okay, great. Okay, that's the side effect of the cancer. What about the second one? And the doctors was like, well, maybe it's because you know, one doctor was like, because your obesity. And then so it was like, no, we can't use that as. I said, well, what about stress? I think stress definitely caused it. No, we can't say that either. I said, well, I don't know what else to tell you, ma'am. I said, I go through this all day long. And what I felt like when I was going through it, I felt like I was in a high stress area. Maybe if we didn't have high stress, I wouldn't have those problems. So I agree with you. I think stress do cause
Oh, yeah.
different illnesses. And as Kam-Meng said, yeah, basically causes our inflammation. So we want to go from stress and then we get inflammation, and now we got cancer cells that's actually activated in our bodies. So yeah, I agree.
Yeah.
So how do all of you reduce your stress?
I live at the beach. I go paddleboarding every day. If you could
I
see
no
it.
longer work. It's kind of
know?
that work.
Well, that's funny because I was just going to mention what you said, Dr. Miller, about if these things increase our stress, it's also possible to decrease it with different methods or exercises. So I actually do meditate, I do yoga. And I feel the minute that I stopped doing those things, actually this week, I've had a terrible neck spasm. And one day also started to feel strange, like a little illness was coming on as well at the same time. And I just felt like, well, you got to stay on your routine because there are constant things in our external environments, like environmental factors, but life things that come up that then cause stress. Stress causes inflammation, which causes illness. So
What
do you do for your stress, Dr. Gary?
I'm not very good at controlling it. You know, I try to take care of my health.
Yeah.
I try to eat well and, you know, not consume alcohol too much and things. But like today was kind of funny. I was eating my oatmeal today, my healthy breakfast oatmeal at 2 p.m.
That's
the first chance I had to eat.
right. That sounds like
the day. So I mean, so it's, you try to do some things well, but then these other factors come in, and it is. But I guess it's strange because you're like, I get very caught up in my work because I think it's very important because I'm trying to help people and, you know, figure out how we prevent diseases and things. And so sometimes like you get so caught up in the work because it's really important, but at the same time, if you get sick because you're not taking care of yourself, then you can't do your work well, you know? So I'm always like, I tell my students and trainees that I want them at their top peak performance. And to do that, they have to sleep and take care of themselves and things. So like I mainly focus on those things about trying to maintain that. But I could definitely use some advice on controlling stress.
so.
Well, you ate oatmeal at 2 o'clock. I had a root beer float at 2 o'clock.
I still haven't had breakfast, so I understand exactly where you are. I'm home all day. But yeah, what I was saying is it's not that I no longer work. I no longer work in that type of field anymore. So I recognize that field was not, I was probably under more stress than the job was giving me stress, because I felt like I was overcompensating for my skills. Whether they were good or bad, I still was doing that. So sometimes I find myself doing that too. And so that can cause stress as well, because sometimes we put stress on ourselves and the job haven't even put those stress on us. So we have to learn how to
I know.
balance that.
I know that.
So
All
yeah.
right, so let's go back to Sambai for a second. So we are you know, because I don't even understand the stresses on people that may live in Ghana, right? I mean, that's one of the things that we're going to try to go study, and we're going to go over there just to meet people and meet, you know, we've now talked to a bunch of people in Ghana and Nigeria about breast cancer, about cancer, about prostate cancer, but still not a lot about their lifestyles and how they live and their daily stresses. And so that's one of the things we want to learn about. But I think a lot of the patients we're going to encounter are metastatic cancer patients, you know, because I think there's so many people who are diagnosed de novo in, you know, in Africa. So their stresses are going to be probably about disease, you know what I mean? Even they're because of this, what they're struggling with. So the fact that they could be very sick cancer patients, is that going to affect our results? Would they be different from somebody?
Well, yeah, I think this is where the work of Lauren's team and doing the interviews and finding that information out, you can start looking at that question. Like, so if they were recently diagnosed and they're asking about what their concerns are, she'll be collecting data that would clearly indicate that that is one of their key stressors. And then in that combination of having the biological samples by having blood from them lets you measure, you know, all of these pathways. Like, so it's like when you think about stress levels, you may have like your cortisol levels measured is one of the things you can do in a blood test.
Yeah.
But that actually turns on hundreds, if not thousands of these molecules in your blood. And so we're like trying to measure all of those different things. So we see activation of stress pathways in our blood tests. And so that's where, like, if Lauren can tell me, she's like, okay, here's the population, and these are the people that had the highest self-reported stress. These had lower self-reported stress. We can see if we see those signals in their blood.
Yeah, you can match them up. Okay, interesting. Very cool.
And maybe help those patients then, through the study and through the findings, maintain a better quality of life because of their possible metastatic diagnosis. Maybe then we can bring in, maybe there's supplements that they could be taking to help with certain things, certain side effects that maybe they're not aware of trying out.
Yeah. So Kam-Meng, you're doing a lot of this work with Dr. Miller, Dr. Gary. What's your typical day like? What'd you do today before you came on this Zoom? Just interesting.
Well, I support Dr. Miller not only on Sambai, but on the whole schools in the research arena, try to elevate research for public health and the public good. So I mean, I joined Dr. Miller on this project because of my cancer background, which I'm very excited to be part of. So my typical day is, I'm not as busy as Dr. Miller, trying very hard to help him to reduce his stress by doing some low
He needs you for that, right?
I mean, Dr. Miller is so successful as a scientist and as Weinstein, so he is under tremendous stress. And
well, every time I talk to you, you're in a different city, in a different country. So
I'm in New York City right now.
And I met your wife. I met your wife in London, and your wife came to London so she could see you because she wasn't going to see you for 2 weeks. Right?
Can I ask a question? I'm sorry. Go ahead.
No, I was going to say that, like, I think that one thing that reduces my stress is my wife, the time I spend with her.
Yeah, she's precious.
That's why, like, she has a remote position, so she can travel with me, which makes the work travel much more enjoyable and things.
That's good. Yeah, she's very precious, but she came over to like to find you. I love that. So I like to be with you. I thought that was great. Dr.
Miller has a very smart and supportive wife, and that is really vital to it. And she's a wonderful person. So I was going to ask about I I don't know so much about breast cancer, but I know that there's some patients have to take tamoxifen for life as a preventive. How, I mean, I heard also there's a lot of side effects.
So
Can you
many
talk
side
a little
effects.
about that?
Yeah, so I had triple negative, so I didn't take tamoxifen. I don't know about you, Danielle, but I hear complaints about it every day. And I think for a while, the protocol was 5 years, and now they're saying 10 years because it's very common with hormone receptor positive breast cancer, which is really the most common breast cancer, but it recurs very readily, especially in Black women. And we're just starting to see that, see that data that for me, triple negative, we don't have a drug to prevent recurrence. And the good thing about triple negative breast cancer is that they say if you make it 5 years, you're good. You're less likely to have a recurrence. Like I had a recurrence, but it was after a year. But I think tamoxifen, we hear all kinds of horror stories from, putting you into menopause and hot flashes and all the bad things that are associated with that. And vaginal dryness. It's a lot of sex talk about tamoxifen and how it hurts your sex life. And so it makes people miserable. And we actually have fans that we give out at events for women who are taking tamoxifen. We have little hand fans.
So
But,
yes, I did not take tamoxifen because I was triple negative as well. But it is for people who back in my time it was 5 years. I'm not sure now that, like they said, they changed to ideas. But my sister actually thinks she was in a study where they tested her using tamoxifen. She's not sure or not because her brain is a little foggy. But she has an eye condition at this moment, and the doctor was like, "Were you on tamoxifen?" She's like, "I'm not sure." He said, "Because if you were, this is a side effect of the tamoxifen." So they're starting to see some type of eye conditions that they're having. And it's almost like they're going into blindness. So there is so much. I've been in so many support groups where women are talking about it, and I usually kind of tune them out, kind of deal, because I can't help them. Not really. I don't tune them out to basically be negative. It's just that it's nothing I can do to help them because I've never been in their shoes. And so, yes, they have so many issues, so many side effects, but I understand once you are finished with that, some of those side effects go away. And so that tremendously helps, but there's so much stuff that people are wondering what could they do to help out with those certain side effects. So yes, a lot that goes on with that.
Yeah.
But there's not only tamoxifen, there's another aromatase, there's another one that actually helps as well. So they actually, back when I got diagnosed in 24 years ago, it was only tamoxifen. Now they've found 2 or 3 other drugs that could actually do the same thing tamoxifen is doing. So they may not take that.
A bunch of different drugs you can take now that are getting approved for tamoxifen. I mean, for preventative. It's a blessing to even have that. But I think that one of the things I think Hope Sambai will identify is, how these things are going to work on the Black cancers. And I know I was with when we were at the AACR conference a few weeks ago, and Sheba was showing me a poster, and she showed me this poster of work that she's doing on triple negative, and then she showed me another thing that she pulled out of her bag. Here's my secret. Don't talk about this yet. Of course, I'm talking about it right now. But she showed me a picture of a white triple negative cell and a Black triple negative cell from a Black woman. And there are little spots on the cells. I'm not scientific at all. Little spots on the cells, but on the white cell, they were all in little clumps, little circles of dots around the cell. On the Black cell, they were just random dots all over the cell. And she was saying how this is like the next phase of her research to show that these cells are night and day different. They don't look anything alike. So when you start talking about drugs for them to cure this cancer, they're different. And I mean, I think if you think about that cell, why are they different? They're different because of exposomics. They're different because of genetics. They're different for all these things. And I don't even know what these little spots are called, but I think that's what we're going to identify in Sambai, that these cancers are different and need different solutions.
Yeah, with the laser focus that this study is going to have, then it allows you to create a different formula. You're able to look at the levels that Dr. Miller was mentioning on such a like zoomed in scale. And you're able to say, this is not looking right. The levels of whatever hormone or whatever is not looking right. We can change the formula of the medication to better suit this population. That's what we hope.
So, Dr. Miller, because we have so many type of pollutants out there, and I know probably in Africa there's probably some different types of pollutants. Are there certain pollutants are we going to basically focus in on? Are we going to try to solve all 40,000 of pollutants while we are doing our research on across this study?
It's an excellent question. So over the last 40 to 50 years, what we focused on are the chemicals that had some evidence were involved in causing cancer. So starting back with the people that were chimney sweeps and they got these very high exposures, to cigarette smoke, which we know has multiple carcinogens in it. But now that we've kind of removed some of those really bad actors or have at least some ways to control them, the concern is like the combined exposure to low levels of hundreds or thousands of things. So what we do with our approach in Exposomics is not to focus on any particular thing. It's the idea of like looking as many as possible to see which ones have the highest correlation to the adverse outcome. So that what are the peaks that have the greatest association with the aggressive cancer? And I don't need to know exactly what it is. I just see it as a peak on my machine. But when I see that corresponding to some very bad cancer outcome, I can then go back to the laboratory and figure out what that peak is. So we are trying to take kind of a broad, unbiased view of all the things that are out there and let the connections to the cancer outcomes be the driver of which ones to focus on.
You're casting a wide net and then kind of filtering it out from there. Yeah.
Do you have any hypotheses on what you're going to see?
I think, again, we do know that there are hundreds of carcinogens. There's things that promote cancer, make it more aggressive. We'll detect a lot of those. Like we'll see a lot of the things that we know. But we're not just looking for them. We're looking,
for anything, yeah.
I think what you'll see is some patterns of things where like individually, these individual things won't reach like a significant level. But when you see 20 or 30 of them together as that cluster, that's when you start seeing that connection. So maybe that cluster are all things that came from a factory that was nearby. And it's all from the exhaust from that factory, or it's related to diesel exhaust, or it's related to high temperature cooking, right? Like when you heat things up when you're cooking, this is like why barbecuing things isn't really healthy for you, because the high temperature creates these molecules that like to attack DNA. And so I think what it is, you're going to see more of these patterns of exposure and not just like single molecules.
So all barbecuing or just when you make it black?
The more heat, it's not as good. So I still like to put things on the barbecue, but I try to eat meats that are healthier, like more chicken breast and less brisket sort of thing. But whenever you have that high level of temperature, it does create, and that's part of the reason like cigarette smoking is so bad, because you're heating the tobacco and those chemicals up, and it makes them more reactive. It's also why vaping's bad, because those little metal coils are heating up those chemicals in there, and then you're pulling them into your lungs. So it's one like high temperature just tends to make molecules more reactive, and carcinogens tend to be reactive.
So it doesn't matter whether I'm eating meat or whether I'm eating vegetables. It doesn't matter because they're all at high temperature. So it's the high temperature that's the problem for the most part.
Well, it's the meats have things in them that are already not as good, and so if I would say if someone liked to grill vegetables, I think you're fine there. Like because eating that high vegetable diet we know is really healthy. And it's not like, oh, well, if you're grilling the vegetables, that's not healthy. No, the vegetarian diet is healthy. So it must be okay to do that. That's very different than, barbecue with, brisket and ribs and pork and all those things, because there's other components of that aren't as good. So,
Right. What about grass-fed filet mignon? I don't know. So yesterday, what did I have for lunch? Hot dogs on the grill. I had this craving. I went in my backyard. I'm gonna die, right? I'm gonna get the kids. But I definitely could eat better. But wow, this is such great information. I'm so proud to be doing this work with Melissa. It's just so awesome to have her, and she probably thought such a great brain trust together to put all these pieces together, and I can't wait to see how this all sort of comes out in the wash, because I know it's going to help the state of cancer. Right? I think
it's a very exciting project, and no doubt I'm honored to be part of the group here and the entire Sambai group, because Melissa really assembled a great team, and it's going to be an exciting few years.
It really is.
So with that being said, have we started doing anything in Work Package 2 with any of the stuff that we're basically so excited to get off the ground with? We started looking at any type of cells, maybe just locally here in the United States, or we're waiting until we get to Africa?
So what we've been doing is it's kind of like optimizing some of our methods for this test and getting new equipment and things. We hope to have several thousand samples from one of our collaborators from Clayton at Hopkins, where it's 2 studies of prostate cancer. Half are from Ghana, half are from the United States. And we hope to get those in a few weeks, and we'll start running them as soon as we get them.
That's awesome. Go ahead, Danielle. Were you going to say something? Okay. Oh, no, that's awesome. So that first group is about how many?
It's a total of about 3,000 samples.
Okay. So they're from patients in just, I think Clayton has great relationships in Ghana, Nigeria, right? Yeah.
It was from one of the studies that he had worked with before. They've actually done a lot of analysis of those people and samples for the genomics and other factors, but they hadn't done exposomics yet. So it's great that they didn't have to recruit new patients. They had blood stored in the freezers, and they're just going to ship it to us.
That's great. So they're going to send it to you, and you're going to run it on your machines, and cool. That's really cool. Really cool. What was Clayton measuring on them already?
I know he'd done a lot of genomic work on them. I think he had some other tumor biology he had done. I think he had some other inflammation marker panel he had done with them as well. And that's what, like, to me, it gets really exciting, is that when you start merging these different types of data together, so the fact that he already has a lot of that, we'll be able to link in,
right,
you see, with these exposures to all these other measures that he's already done.
Awesome. Well, thanks for this today.
Thank you so much for your time.
Yeah, I know you're really busy, and we have, we're so excited to do this and share this information with our audience, and we're hoping that we'll keep growing our audience. So share this with your friends if you're watching. Share this with your friends, and we will come back in a month and do it again with our next work package partners. But we are so excited I want to come visit you guys in New York and spend a day.
I do have one question. For those not involved in the study and want to know more about Exposomics, is there anywhere that they can go learn more?
Yes,
fairly new.
We have a website for our center that we have here at Columbia that has a lot of information on it. We recently did a podcast with the Mayo Clinic where they interviewed me and a colleague of mine about Exposomics. It was a again, that was, it's just a nice summary of some of the general workflow that we do.
Oh, that's great. We'll post that.
Yeah, and so we can share those links with you to share with.
Great. We'll post that for sure with our audience. Yeah. And we'll come back to, we're kind of going through every work package, and then I think after we do all of them, we'll come back and kind of pull you back.
Check in.
With everybody to say, okay, here's where we are now, right? And so by the time we get ready to go to our next Cancer Grand Challenge meeting in March, right? So yeah, so thank you for your time today. And
all right, you're very welcome. Great seeing you.
We'll see you soon. See you on the. Okay, thank you.
Thank
Bye-bye.
you.